Assalamu alaikum everyone. My name is Abeduzzaman Anon. I'm a research assistant at BIOICON Academy, and today I will be presenting a paper titled Proteome Signature of Alzheimer-Like Phenotypes in Frontal Cortices from young and old individuals with Down syndrome. This paper is published in Molecular Neurobiology in 2025 by Domenico Gresso and Tramutola and colleagues from Sapienza University of Rome, also collaborating institutions across Europe and the United States. Before I begin, I want to set the stage for why this paper matters. We are looking at a question that sits at the intersection of genetics, aging, and neurodegeneration. So why do nearly all people with Down syndrome develop Alzheimer's disease by the age of 50? And can studying their brain at the protein level tell us something fundamental about how Alzheimer's begins in everyone? So let's start. Let me give you a brief roadmap of this presentation. We will cover here the scientific background and why this study was necessary, the materials and methods in details, including the proteomics technology and the bioinformatics pipeline, the results group by group with a full explanation of every major figure and diagram, the discussion and conclusions, also the limitations and future direction. So in this slide, it is the introduction of Down syndrome. So Down syndrome, also known as trisomy 21, is the most common genetic cause of intellectual disability in humans. It occurs when a person carries an extra copy of chromosome 21, referred to throughout this paper as HSA 21. This happens in approximately 1 in every 1,000 live births. So the clinical picture of Down syndrome is diverse. Common features include facial and skeletal abnormalities, congenital heart defects, hematological complications, gastrointestinal abnormalities, also critical neurological disorders. Among these, the most devastating is the extremely high risk of developing early onset Alzheimer's disease. Now, a question. What makes this biologically remarkable? In the concept of gene dosage imbalance, having 3 copies of chromosome 21 instead of 2 means that all genes encoded on that chromosome are expressed at higher levels. So this extra expression does not affect only HSA 21 located genes. It also triggers a cascade of genome-wide transcriptomic alterations, affecting genes on all other chromosomes too. So these are the common features, as I have told you: facial abnormalities, congenital heart defects, duodenal atresia, skeletal disorders, transient abnormal myelopoiesis, and neurological disorders. So on the next slide, why proteomic analysis? So here is a critical gap that this study addresses. Prior work has focused heavily on transcriptomics, studying which genes are turned on or off. But gene expression does not always translate directly to protein production. You can have high mRNA for a gene and still have normal or even low protein levels because of post-transcriptional regulation, epigenetic modifications, or protein degradation. So proteins are the actual workhorses of the cell. They carry out every function like metabolism, signaling, structure, defense, and so on. So if we want to understand what is going wrong in a Down syndrome brain, we need to look at the proteins directly. This is exactly what this study does, as I have shared you with the research paper. Using a technique called shotgun proteomics is mentioned in here. The researchers analyzed 4 human groups from post-mortem frontal cortex tissue. Number 1, young individuals with Down syndrome. They are called, as mentioned in the article, DS, age-matched young healthy controls, called CTRY or control youngs. Older individuals with Down syndrome who also had confirmed Alzheimer's disease. They are mentioned as DSAD, age-matched older healthy controls, called CTRO, controls old. And additionally, they used a mouse model of Down syndrome. They are mentioned as TS66YAH at 2 times point, like 3 months and 9 months of age. So in this slide, the methods table is mentioned. Let me walk you through the methods carefully, because understanding the technology is key to understanding what the result actually means. So in here, human brain samples like post-mortem frontal cortex samples were obtained from 3 established brain banks. Number 1, University of California Alzheimer's Disease Research Center. Number 2, Eunice Kennedy Shriver, NICHD Brain and Tissue Bank. And number 3, University of Kentucky Alzheimer's Disease Center. A total of 24 human brain samples were used, 6 per group. As you can see in here, 6 per group. And the Down syndrome group had subjects under 44 years of age. The DSAD group were over 55 years old and had confirmed Alzheimer's neuropathology. The young controls were up to 45 years old and the old controls were over 45. Sex was approximately balanced, 4 males and 2 females, or 2 males, 4 females in most groups, with the DSAD group having 2 males and 4 females. Importantly, statistical tests confirmed no significant difference in post-mortem interval, the time between death and tissue collection, across these 4 groups. This is critical because delays in processing can degrade protein quality and confound results from those samples. And if I talk about mouse samples, the TS66YAH mice were used as a validated model of Down syndrome. These mice carry a partial triplication of the mouse equivalent of HSA21. They were sacrificed at 3 months and 9 months of age, representing young and older adult stages, with 6 mice per group, equally split by sex. And now let's talk about protein extraction, the analytical approach. Brain tissue was homogenized in RIPA buffer, RIPA buffer, a standard lysis solution. It contains protease and phosphatase inhibitors to prevent protein degradation. Samples were sonicated on ice and then centrifuged at 16,000 RPM to remove cell debris. Total protein concentration was measured using the BCA assay, a standard colorimetric method. So the proteomic pipelines of NLC-HDM-SE, NLC-HDM-SE, as mentioned in here, the shotgun proteomics. So this is the heart of the methodology. The team used a technique called label-free nanoscale liquid chromatography combined with high-definition mass spectrometry in expression mode, abbreviated as NLC HDMSE. So let me break this down step by step. FASP trypsin digestion, that mentioned in here, FASP protocol. So proteins cannot be analyzed whole by mass spectrometry. They are first digested into smaller fragments called peptides using an enzyme called trypsin. Following the filter-aided sample preparation FASP protocol. This standardizes digestions and removes detergents. Internal standard addition before injection of 300 femtomoles of yeast enolase, a well-characterized protein from baker's yeast, is added to every sample. This acts as a spike in internal standard for qualification, normalization, ensuring that difference between samples reflect biology, not technical variation. Also, let me talk about nanoscale liquid chromatography, NLC. So, the peptide mixture is separated by passing it through a tiny reverse phase column, like 75 micrometers in diameter and 150 millimeters long, using a 120-minute gradient. This separates peptides by their chemical affinity, sending them into the mass spectrometer in a controlled sequence. After that, ion mobility separation, here mentioned as HDMSE. This is the most advanced part of the pipeline. The mass spectrometer uses a technique called ion mobility separation in here, which adds an extra dimension of separation, separating peptides not just by mass and charge, but also by their 3D shape as they travel through gas. This dramatically increases the number of peptides that can be identified as and quantified simultaneously. So next, data independent acquisition. Like unlike traditional proteomics, HDMSE uses a data independent acquisition, like DIA strategy, meaning the instrument scans all peptides in the sample continuously without pre-selecting specific targets. This produces comprehensive, unbiased proteome coverage. Next, each sample was run in three technical replicates, meaning 3 separate injections per biological sample to ensure reproducibility. After that comes data processing and statistics. These raw data was processed using Protein Links Global Server, PLGS software. Proteins were identified by matching measured peptide masses to the UniProt human and mouse databases at a false discovery rate of less than 1%, controlled using a target decoy database strategy. For a protein to be called differentially expressed in here, it had to meet 2 criteria. Number 1, it had to appear in at least 2 of the 3 technical replicates. Number 2, it had to show a fold change of greater than or equal to plus 30%, meaning at least 30% more or less than the comparison group, with a p-value below 0.05 after Benjamini-Hochberg correction for multiple testing. Sex, age, and postmortem interval were all included as covariates in the statistical model to control for their influence. After that comes bioinformatic analysis. Differentially expressed proteins were then analyzed using UniProt database for protein characterization like chromosomal location and subcellular localization. IPA here means ingenuity pathway analysis. It reflects the identity of which biological pathways and disease networks were affected. IPA uses Z-scores to indicate whether a pathway is activated. That means positive Z-scores means pathway is activated, and inhibited, like negative Z-scores means the pathway is inhibited. Then string software. String visualize protein-protein interaction networks and identify functional clusters. Also Venn diagram to compare overlapping proteins across groups. So now let's discuss about results, diagram by diagram. Young Down syndrome versus young controls. The early genotype signature is explained in this slide. The comparison between young Down syndrome individuals and young controls identified 432 differentially expressed proteins. The volcano plot figure 1a. This figure represents each detected protein as a dot, these dots. The horizontal axis represents fold change in expression, and dots to the right are upregulated in Down syndrome, and dots to the left are downregulated in Down syndrome. The vertical axis represents statistical confidence. Higher dots are more statistically significant, red dots are upregulated, and blue dots are downregulated. The scatter of dots reveals that the dominant trend is strongly towards upregulation. 301 proteins, like 69.6%, were increased, and only 131, like 30.4%, are decreased. This tells us that in the young Down syndrome brain, the cellular machinery is in overdrive. Now the violin plot, figure 1b, the chromosomal distribution. This plot shows which chromosomes encode the altered proteins. Critically, despite trisomy 21 being the cause, only 2% of differentially expressed proteins were encoded on HSA 21. The largest contributions came from chromosome 17, 11, and 12. This is the trisomy paradox. The extra chromosome acts as a master switch, disrupting protein expression across the entire genome. So, the HSA21 encoded proteins that were altered include SOD1, S100B, APP, CBR1, and components of ATP synthase, all consistent with previous Down syndrome brain studies. Next, the pie chart. In figure 1c, you can see the subcellular localization. This pie chart breaks down where in the cell the altered proteins are normally found. The largest slice, 38.6%, belongs to the cytoplasm. But the most striking findings is that 19.5% of the differentially expressed proteins are mitochondrial proteins. This is disproportionately high and tells us that mitochondrial biology is the primary target of genotype-driven dysregulation in the young Down syndrome brain. A small but meaningful 0.25% are synaptic proteins, hinting at early disruptions in neuronal communications. And on the right side, the bar graph, figure 1b, represents canonical pathways. This graph lists biological pathways on the vertical axis and their activation z-score on the horizontal. Bars extending to the right, positive z-score, indicate pathway activation, and bars to the left here, that indicates inhibition. Here, oxidative phosphorylation means the cell's primary ATP generating process is surging. Respiratory electron transport, that is closely related, also strongly activated. Synaptogenesis is new synapse formation that is being driven upward. The mTOR signaling means a master growth regulator is activated. The NRF2 mediated stress response represents the cell's antioxidant defense is engaged in here. The mitochondrial dysfunction, this negative z-score is interesting. It means genes associated with mitochondrial dysfunction are present but are being partially compensated or countered in here, in this case. Sirtuin signaling, like longevity related signaling, that is suppressed in this disease. The pattern suggests a young Down syndrome brain that is metabolically overreactive but stressed, running hot and trying to compensate. So after that, amyloid pathway network, we can see in here the network diagram of figure D.1, this figure. This IPA network diagram shows the molecular relationship between differential expressed proteins associated with amyloid production and microtubule instability. The central nodes include APP, amyloid precursor protein, ERK 1/2, ERK 1/2, P38 MAPK, this one, P38 MAPK, and APOE. The arrows and connections shows how these proteins interact to drive amyloid fiber formation, which receive a positive z-score of +2.2. Critically, APOE showed a +1.7 log-fold increase in young Down syndrome individuals. Like, it makes it one of the earliest molecular markers of Alzheimer's disease. This is remarkable because APOE overexpression is typically associated with late-stage Alzheimer's disease, not with young adults. So the string network of this figure H1H, this string diagram is a complex wave of interconnected circles, each representing a protein with connecting lines indicating known or predicted interactions. The analysis reported 396 number of nodes, that means proteins, 6,919 ages, that means this amount of interactions between proteins, and average node degree of 34.9. That means each protein on average interacts with nearly 35 other proteins. Protein-protein interaction enrichment p-value is less than 1.0 into 10 inverse 16. The network is far more interconnected than would be expected by chance. This value means this description. The most prominent clusters were carboxylic acid metabolism, oxidative phosphorylation, and the proteasome, confirming the themes of energy disruption and protein degradation stress. On the next slide, we move to the older groups. DSAD versus old controls in the Alzheimer's stage signature. So in here, the comparison between individuals with Down syndrome and Alzheimer's disease versus age-matched controls identified 281 differentially expressed proteins, fewer than in the young comparison from the previous slide. The upregulation trend persists at 67.6%, but the total number of altered proteins is declining, suggesting that the proteome is exhausting itself. In this slide, the volcano plot figure 2a, this plot represents a structurally similar to the previous one, but you will notice a smaller spread of data points. The overall volume of significant changes is shrinking, consistent with declining cellular activity as Alzheimer's disease takes hold. The chromosomal distribution of HSA21 proteins now account for only 1% of total differential expressed proteins. The dominant alteration shift towards chromosome 1, 11, and 17. The genome-wide disruption continues, but reorganizes itself as the brain ages into Alzheimer's disease. On this slide, the pie chart C, subcellular localization. This is a fascinating comparison with the young groups chart. Mitochondrial protein drops from 19.5% to 12%, while membrane-associated proteins increase to 25.1%. This shift signals that the late-stage disease is less about energy production failure and more about membrane integrity and synaptic structural breakdown. Synaptic proteins also slightly increased to 0.4% of the total differential expressed proteins. Pathway changes means the NRF2 antioxidant response flips negative. Insulin receptor signaling is inhibited. The TCA cycle and glycolysis remain positive, but with less force. More intriguingly, autophagy pathways show a positive Z score. That means counterintuitive, but it matches established findings in Down syndrome. The early stages of cellular cleanup are initiated here, but the later degradation steps often fail. It is as if the brain is trying to take out the trash, but the rubbish truck never arrives. This types of situation is occurring in here. So after that, the string network figure 2G. In the old brain, the network shows 209 nodes and 1,381 edges, a denser relative connectivity than in the young comparison, indicating that the surviving altered proteins are now highly coordinated in their dysfunction. Primary clusters center on protein folding and oxidative phosphorylation, the brain's last-ditch attempt to manage protein misfolding and maintain energy. Perhaps the most clinically important findings in here is that a 62% overlap was found between DSAD proteome and established databases of sporadic Alzheimer's disease. This is the molecular proof that Down syndrome associated Alzheimer's disease and classical Alzheimer's disease coverage on the same biological endpoint. So what is common across young Down syndrome and old Down syndrome Alzheimer's disease? This Venn diagram shows three overlapping circles. Number 1, 271 differentially expressed proteins unique to the young Down syndrome group. Then 161 differentially expressed proteins. These are shared between both Down syndrome groups, young and old. After that, 121 differentially expressed proteins unique to the DSAD group. So these 161 shared proteins represent the core genotype signatures of Down syndrome. Molecular changes that are driven by trisomy 21 itself and persist across the entire lifespan, regardless of age or Alzheimer's status. So after that, heat maps. We can see many heat maps in here. These heat maps display specific proteins and their expression values, shows as color intensities across the two comparison groups side by side. So in here, D.1, the glycolysis, this represents ENO2 and PKM are shared. These two genes are shared across both young and old Down syndrome groups, maintaining consistent upregulation. In the next one, synaptogenesis. In that diagram, synaptic proteins like CA-DNA21 and YES1 are altered in both groups. In the third heat map, mitochondrial dysfunctions, shared proteins include ATP, 5F1B, and DNM1L, then VDAC1. These are core mitochondrial energy and structure proteins. After that, the NRF2 stress response heat map. This one shows that functional switch. This heat map reveals the functional switch. In young Down syndrome, NRF2 related proteins are upregulated. You can see that in here warm colors, and in DSAD they flip downwards. The brain's antioxidant defense actively activates early, then fails with age. In number 5, chaperone mediated autophagy. This one represents shared proteins include HSP90AB1 and GFAP hallmarks of stress response. In the next one, AMPK signaling. Here, energy sensing proteins remained shared. After that, the PCA cycle heat map. This one showed key metabolic enzymes that are consistently altered. And on the last heat map, oxidative phosphorylation, 4-OXPHOS proteins like COX5A and COX7A2, they showed shared patterns. So now let's move on the next topic. What happens as Down syndrome brain age into Alzheimer's? When we directly compare older Down syndrome individuals to younger Down syndrome individuals, we capture that the pure aging effect within the Down syndrome brain. This comparison identified 902 differential expressed proteins, the largest number in this study. And here, in the critical reversal, 63% of this protein are decreased, while only 37% are increased. In the volcano plot, you can see the distribution of dots is now shifted to the left side. The dominant trend is down-regulated. This Down syndrome brain is no longer overreactive. It is mainly shutting down. And in the figure B, pathway analysis, here you can see the key suppressed pathways that includes oxidative phosphorylation, like the value is minus 1.85. Respiratory electron transport, PCA cycle, NRF2 redox response. Then chaperone mediated autophagy. Lastly, ERK MAPK pathways. So the amyloid formation pathway also shows a negative Z score of minus 1.63. Not because amyloid is decreasing, but because the proteins here driving amyloid production in the young Down syndrome brains have already done their damage. The pathological hallmarks are already in place. The machinery that built them is now exhausted. Mainly, I am indicating to brain. This string network, figure 4E mentioned in the article, this network highlights a dense interaction cluster of 108 differentially expressed proteins involved in carbon metabolism and 57 differentially expressed proteins linked to mitochondrial ATP synthesis. This is essentially a portrait of brain's power supply collapsing. A coordinated failure of energy production that likely underlies the cognitive decline of Alzheimer's in Down syndrome. So on the next slide, normal aging versus Down syndrome aging. Mainly, it represents the premature aging proof. The Down syndrome of old and young neurotypical controls found 760 differential expressed protein with 65.7% downregulated, mirroring the DSAD versus Down syndrome patterns. This confirms that protein decline is a universal feature of aging brain. Most strikingly, the aging normal brain initiates amyloid-related process, changes in APOE, APP, SOD2, and SYNGI, the same proteins that are already altered in young individuals with Down syndrome. In other words, what happens here is that naturally to the brain at age 60 to 70 in a healthy person has already happened in a 25-year-old with Down syndrome. This is the strongest quantitative proof of Down syndrome as a premature aging. So in this slide, the information represent what aging pathways are, why aging pathways are universal. The comparison between aging in Down syndrome and aging in controls showed an 80% overlap in differential expressed proteins. But it confirms that the core molecular program of aging is fundamentally the same. Shared proteins include MAP, tau, SOD2, GAP43, SNAP91, OPA1. These are all established markers of neurodegeneration. The difference is severity. The Z-scores, statistical significance, and magnitude of pathway suppression are all much greater in Down syndrome aging group. The trisomic genotype acts as an accelerator of a universal program. The same road, but driven at 3 times the speed. In this slide, the combined effect of genotype and aging is mentioned. When all 4 comparison groups are analyzed together, only 150 differential expressed proteins, 12.3%, are influenced by both genotype and aging simultaneously. These include CAM2K, MAP1B, PGAM1, ENO2, MAPK1, GAP43, and BUFS5. These proteins are highly sensitive to chromosome 21 triplication and to the aging process. The walled cloud that represents the size of each protein name reflects how frequently it appears across comparison group. The largest terms CAM2K, CAM K2A, ENO2, GAP43, MAP1B. These are your most consistently dysregulated proteins across the entire dataset. These are your primary candidates for biomarkers and therapeutic targets. The key conceptual finding in here is genotype and aging work is in opposing direction. Genotype tends to activate pathways, aging tends to suppress them. But together they converge on the same pathological endpoint, Alzheimer's disease. So now the mouse model, confirming across species. The TS66YAH mouse model analysis provides critical cross-species validation. At 3 months of age, the mouse proteome showed a massive 95% upregulation of proteins, mirroring that the proteomic surge in young human Down syndrome individuals, the energy pathways like oxidative phosphorylation plus 5.3 and glycolysis like plus 3.1 were among the most activated, as was the NRF2 stress response and neurotransmitter release. The Venn diagram shows that 149 orthologous proteins, like minimum 21.9%, overlap between young human Down syndrome and young TS66YAH mice, confirming that early genotype-associated pathological mechanisms are conserved across species. At 9 months, the same 95% upregulation trend persists in mice, unlike the reversal seen in the human Down syndrome and Alzheimer's disease. The overlap with human Down syndrome Alzheimer's disease drops to only 78 protein, like 9.3%. But this divergence occurs because mice don't develop the severe chronic Alzheimer's pathology seen in humans, partly because mouse APP is processed differently. The mice capture the genotype-driven metabolic surge, but not the human experience of decades-long neurodegeneration. So now we have arrived to discussion part. The discussion section synthesizes these findings that I have discussed into 4 major conclusions. First, the trisomy paradox. The trisomy paradox. Approximately 90% of differentially expressed proteins in this study are encoded on disomic chromosomes. HSA21 does not just add extra copies of its own genes, it rewires the expression of the entire genome. The weak correlation between mRNA and protein levels observed in this and other studies tells us that the disease cannot be understood from transcriptomics alone. Proteomics is not optional, it is essential. Secondly, from proteomic storm to exhaustion, the young Down syndrome brain experiences what the author calls a proteomic storm. It is a massive upregulation of proteins. particularly in energy and proteostasis pathways. This is the brain working overtime to compensate for the trisomic imbalance as Alzheimer's develops. This compensation fails and the proteome collapses. The overall number of differential expressed protein decreases, protein expression falls, and pathways shut down. Thirdly, proteostatic failure as the central mechanism, the loss of the brain's ability to maintain protein balance, to fold proteins correctly, and clear misfolded ones. Also, regulate proteins turnover. This is the central molecular feature of Down syndrome-linked Alzheimer's disease. This failure drives amyloid beta deposition and tau tangles, which are established long before clinical symptoms appear. The elevated APP and APOE levels in young Down syndrome individuals indicate the process begins in the third decade of life or even earlier. Fourth, mitochondrial failure and premature aging. Mitochondria are the most heavily affected organelles in Down syndrome brains. The young Down syndrome brain shows compensatory metabolic surges. The old Down syndrome brain shows a catastrophic crash. These findings suggest that a 62% overlap exists between the Down syndrome, Alzheimer's disease proteome, and sporadic Alzheimer's disease databases. Is the strongest molecular evidence that Down syndrome represents a genetic form of Alzheimer's disease, not merely a risk factor or association, but a true biological coverage. So to summarize the main conclusion of this discussion, trisomy 21 establishes a unique proteome signature in the frontal cortex from early life, characterized by energy surges, mitochondrial stress, and early amyloid priming. As Down syndrome individuals age into Alzheimer's, the proteome undergoes a dramatic reversal from upregulation to widespread protein decline. Normal aging and Down syndrome aging share the same molecular pathways, but Down syndrome accelerates these processes by decades. The TS66YAH mouse model faithfully replicates early genotype-driven proteomic changes, validating it as a useful tool for Down syndrome research. Key proteins like APOE, APP, MAPT, GAP43, VDAC1, SLC1A3, these are identified as persistent molecular landmarks of the Down syndrome to Alzheimer transition and represent priority targets for further biomarkers development and therapeutic interventions. So now, limitations acknowledged by the authors. So these limitations include small sample sizes, like only 6 individuals per group. So this is inherent to post-mortem brain studies and limits statistical power. The analysis was restricted to the frontal cortex. Other regions like hippocampus may tell a different story. Post-translational modifications and protein-protein interactions were not fully captured by this bottom-up proteomics approach. Low abundance proteins, membrane proteins, and isoforms are underrepresented here by current mass spectrometry technology. BRAC staging data, which classified Alzheimer's disease severity, was unavailable It prevents correlation of proteome findings with disease progression scores. Despite these limitations, this is the first proteome-wide characterization of the human Down syndrome brain across aging and Alzheimer's disease development. So this is a landmark contribution to our understanding of the molecular mechanism driving neurodegeneration in this population. So lastly, I want to say that Down syndrome has long been viewed as a childhood condition, something that affects development, learning, and early life. But this paper reframes it as a lifelong neurological trajectory. That means one that begins with a genetic overdrive in early adulthood. It progresses through a compensatory proteomic storm and ends in the same molecular catastrophe as Alzheimer's disease, just decades ahead of schedule. By mapping this trajectory at the proteome level, protein by protein and pathway by pathway, this study gives us something that was previously missing, a molecular timeline of how Alzheimer's begins in a genetically predisposed brain. That timeline may hold the key not only to helping individuals with Down syndrome, but to understanding the ultimately preventing Alzheimer's disease in all of us. So that's all from my side. Thank you very much. And now I am happy to take any questions for further discussion. Please, everyone, if you have any questions, you can ask me. If you guys don't have any questions, there were some questions previously asked, so I will discuss those questions. Like number one, why was the frontal cortex specifically chosen for this study? As we have seen in the article, the frontal cortex is one of the brain's region most severely affected by Alzheimer's disease. It governs higher cognitive functions like memory, decision making, and personality. Since Down syndrome individuals develop Alzheimer's disease-like pathology in this region early, that's why it is the most relevant tissue for studying the Down syndrome to Alzheimer's disease transition proteomically. That's why that's why to discuss here, the frontal cortex was specifically chosen. If you guys have any questions, you feel free to ask me. I will try to answer those questions as well. So the next question came, why is proteomics more informative than transcriptomics for this study? Well, to be honest, mRNA levels do not always reflect actual protein levels. Post-transcriptional regulation, epigenetic modifications, and protein degradation can also decouple gene expression from protein abundance. Since proteins are the actual functional molecules in here, in these cells, measuring them directly gives a more accurate picture of what is biologically helping or happening in the Down syndrome brain.